Recording. I guess I should advance the slides. All right, so we'd gone through Darwin's theory of evolution. Yeah, that's really important for understanding how come life is so diverse on Earth. And that goes back to that picture I was just looking at and talking about before I turned the recorder on. That's what amazes me, is how many different types of life there are on the planet. And that's, like I said the other day, just a little bit of the life that's been on the planet. During the Permian 200 million years ago, did they show that on that chart? I didn't look at that. So 200 million right here. This mass extinction. Where's my pencil? Oh, I can point. There we go. This one right here is the one I'm talking about. Kind of on the right side of the circle. You can kind of see where they're showing it there. There was a mass extinction that occurred during that time, and it killed off 95% of all marine species. Remember, I was telling you about that. It had a lot to do with the way the continents were forming. So to me, it's always kind of interesting to think about how that all happens and how that all sort of happens according to Darwin and his theory. And remember, we talked early on about the difference between a hypothesis and a theory, right? There's no mistaking the difference between the two. A hypothesis is something that you believe is going on. A hypothesis is something that's repeatedly tested. Hypothesis is something that maybe not everybody quite ascribes to, even scientists do yet. Maybe they're still debating parts about it. But there are certain hypotheses that have been tested and looked at so many times, remember, that they become theories. The rotation of the Earth on its axis is simply a series of hypotheses confirmed through observation and different types of observations through time that became a theory, the theory of planetary rotation. However, every day the sun does come up, substantiating that theory, right? So a theory is a collection of hypotheses. In other words, an idea has been proven not to be wrong many, 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 many times. And so Darwin's theory of evolution is it's not a hypothesis. It's a collection of millions, well, maybe not millions, many, many, many hypotheses that have been shown to not be wrong. Remember, in science, we never prove anything true. We just prove it's not wrong. So we either refute, which is like, I don't think so, or we support the hypothesis. And everything we've seen about Darwin's theory of evolution is laid out on this screen has been supported by hypothesis after hypothesis after hypothesis. There's no doubting Darwin's theory of evolution based on this mechanism of natural selection, okay? There may be some of the details that still need to be worked out, but overall, Darwin, pretty smart guy, had it figured out on the biological level very early on that the way it works in evolution, that the critters that survive, that the organisms that extend out that branch that I was just showing you guys in that other picture, those are the ones that reproduce more, right? So at the bottom, that differential reproductivity tells you what I'm trying to emphasize by that. Evolution is not the Grim Reaper. Natural selection is not the Grim Reaper. It's not survival of the fittest in the sense that it's just this big fight and everything's killing everything. I mean, yes, that is what's going on, but what drives evolution is the fact that you can reproduce more than somebody else. It's not that you die off more than somebody else. Now, I know that's a subtle difference, because if you think about it, the earlier you die in life, if you suffer very young mortality, then obviously, especially before you get to the age of sexual or before the age of the ability to reproduce, then obviously you're not leaving behind any offspring, and that is part of it, yes, no doubt. But really, what it's about is everybody's leaving offspring. So let's assume just the population we're talking about is sexually mature or able to reproduce, even if they do it asexually. Okay. Those that have some little edge, maybe they reproduce faster, maybe they can reproduce on less food, maybe they've learned how to live on an alternative food. But whatever the case may be, whatever this advantageous, right, that would be an advantage, of course, if you could live on a different type of food, because less competition than all your buddies out there, right? So whatever that might be, that advantage allows you to be well, not stronger, fitter, yeah, to be more fit, and more fit means it's precisely defined in evolutionary science. Fitness is basically your genetic contribution to the future versus everybody around you. Okay, so it's basically how many offspring I leave versus how many offspring you leave, and whoever leaves behind the more offspring throughout evolution has been the lineage that's survived. Okay, because it's all about leaving. But now every once in a while an organism has taken a detour and produced lots of offspring, but couldn't keep up with the change in the environment and gone extinct. That's the truth. But when it comes right down to it, if you're competing at the basic level, and it is a competition, but it's not a combat, okay? It's a competition of reproductive rates. The more you reproduce, now if you can live a long time and reproduce a lot, you got the, what is that called, the nexus? You got the synergism there, you got the combination of the best of both factors, right? So don't let anybody ever raise doubts in your mind about this theory of evolution, not just a hypothesis. All right, any questions on that? Okay, we jumped through the different kinds of, plankton. Aren't those neat? God, look at that. What was it? Von Leeuwenhoek, when he looked into a microscope and saw protozoans. I'm sorry if I repeat my stories, but he looked into the microscope and he saw these microscopic critters. Now, microscopes were getting to the point back then where you could see them, and he made some quote like, "No more beautiful sight has my eye ever seen," or something like that. Something very blah blah, right? Something very ho ho ho. But man, I tell you, I remember when I was a kid in, oh gosh, 6th grade maybe, doing the same thing with a drop of pond water and just being totally mesmerized by following these little critters around, you know. And what we're talking about, of course, are the plankton. And remember, plankton isn't a type of critter. It'll be used very often in that context, and so it's confusing, okay? But there are different, plankton is a lifestyle. It's the floating lifestyle, remember? Many oftentimes, no doubt, because you're a floater, you're small, right? And you can be bacteria size and virus size, which is really small. Now, here's where the word plankton becomes kind of confusing. In the holoplankton, notice that you're always planktonic. You're always floating, all right? So that's where the confusion comes about, because sometimes plankton are organisms that are always floaters, like these ones up above I told you in class. These would be the plankton. These would be the floaters. Many of the, well, looking in there, and I'm not an expert on plankton, but looking at those look like the organisms that always stay as plankton. You've heard about a krill, for example, whales, filter feeders down in the whales, down in the Southern Ocean a lot. They feed on krill. Krill are little miniature shrimp. I think these might be krill. Again, not being a plankton expert, so some of you may know better than I. That's a copepod in the lower right. This is a weird thing called a salp. It's actually a mucus net. This is the critter. So it tows this mucus net and it catches stuff with it, feeds upon it. If you're a small cephalopod, if you're a small squid, you'd be planktonic. Or like the eels, even the eels I studied in college, that was their larval stage. Cool name for this, by the way, I didn't tell you guys in class. We call the larval stage of an eel. Oh, by the way, eels, tarpon, and bonefish all have the same larval stage. It looks like this. Now, bonefish, tarpon are fish, right? But that tells us that eels and fish share a very recent common ancestry, at least those kinds of fish, okay? This is called the Leptocephalus, there we go, Leptocephalus larvae. So those are planktonic, and then sometimes those planktonic things change, right? They grow, and then they're no longer planktonic. So you can be a plankton, or you can live the life of a plankton. I guess you can be a pirate or live the life of a pirate. That's a really bad analogy, but I think you get the idea. Any questions on what's a plankton? What's a plankton? Okay, good deal. And a cool word. Use that today. Leptocephalus. Cephalus is head. Lepto. What's lepto? You know what a klepto is. Lepto. What's a lepto? Lepto means, Like leptospirosis. There you go. Leptospirosis, spiral, maybe. You guys get internet connection. Go there and tell me look up what is lepto mean. What does lepto mean? Small, narrow. All right, so this is a very thin leaf-like, yeah, that's right. Okay, leptocephalus. So what you're seeing is a head and then the body behind it, and those things I pointed to are going to become the vertebrae, but this thing is like paper thin and transparent. So it's a plankton because it's floater. Now, why do you think it's transparent? What do you think the significance of being transparent would be in a really tall, small little larval form like that? Yeah, you can't be seen, right? Can't see me. Can't see me. There you go. All right. Nekton is when you swim around. Now we're going to talk more about fish in some other chapters too. So we'll come back to those. I love fish. I can talk about them all day, but we do have to talk about the other critters. Remember, sometimes in isolated areas, critters get really big. Like this guy here, this huge isopods. Did you guys look up isopods online? Isopods aren't very big. They're typically not very big. But this is a big one. If you live in those areas that are sort of by yourself, sometimes you get really big. Sometimes, I think we talked about key deer. Sometimes you get really small. Down in the Florida Keys, there are white-tailed deer that are miniature versions, and that's as big as they get. It's one of those things where sometimes if you live in a small area, you have few resources, so you don't grow very large. Or sometimes if you live in an isolated area, you have access to all the resources. So if you're you can grow much larger. Okay, so it's kind of a delicate balance there. But these critters that we're looking at are the ones that live on the bottom. Now they might be fish. Some fish live on the bottom, so we would call them benthic species. Doesn't mean they don't swim. That flounder, if you tickle him, whoop, whoop, he'll come swimming up out of that sand, shaking that sand off, and he'll swim sideways. Swim side, and then he'll settle back down. And then what they do once they settle back down, see if I can pull my hands the right day. Here we go. They go and they wiggle, and as they wiggle, they bury themselves in the sand again, only leaving their little eyeballs stick up. And conveniently their mouth. Conveniently. So here comes little Nemo. Whoop! Don't even know you're there. So not only can I hide from things, I can actually use it as ability to ambush. Now, very, very cool. Very sharks. There's some species of sharks that can feed upon these fish that bury themselves in the sand. All right, they bury themselves in the sand, and the shark can't see them. But if you've ever seen the front, I don't even know if I have a picture. I think we talk about this later on. But if you've ever seen the underside of the nose of a shark, that's an area called the rostrum. Could be any species actually. But if I was looking at the underside, and so I say here's the mouth, there's little black dots all over the underside of the nose of a shark. If you've ever dissected a shark, I know your teacher talked about them. You've seen I'm gonna dunk on it. Here we go. Okay. What they are in just a minute. I'm gonna show you a link. Link. Okay. Here we go. I'll put it in the chat, and then you just click on it. Look at those. Look at those things. Those little pits, I guess, right? Now you see what they're calling on the right-hand side. It's obviously some name applying to, something to do with some fellow named Lorenzini. But Lorenzini's a guy who described these things. Okay, and these are called ampullae of Lorenzini. They look like little blackheads. So this looks like a shark going through puberty, right? Okay, but they're not blackheads. What they are is they're electrical receptors. So these sharks will swim across the bottom, right? Everything gives off an electrical field. Call it aura if you want to, right? Everything that's alive generates a small amount of electrical current. Some species like electric eels and that kind of stuff, which is really not an eel, by the way. But they can use that, right? Generate a lot of current. But even you generate current. So that shark uses these ampullae of Lorenzini. Is it pulling up when you guys click on it? You guys see what I'm talking about? Yeah, so they use these things to sense electrical fields. So they'll swim very close to the bottom across the top of these flatfish that are buried in the sand, and they'll detect them by the electrical field that that fish or worm or mollusk or whatever they're feeding upon, anything alive down in the sand. Is that neat? All right, now knowing that you know that, there is this phenomena that occurs many times when a shark is swimming up to a shark cage. And a shark cage is made of aluminum. Oftentimes it's a little lightweight, strong metal, blah, 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 right? So they immerse it in seawater because you're going to find sharks often in seawater. So we've got salt water, we've got aluminum, we've got a metal. We immerse you inside of this cage. We tie a big old chunk of bait out there. Usually it's a big old piece of a horse or something like that. The shark comes swimming up. At the last minute, the shark closes these membranes over its eyes. These membranes are called nictitating membranes. Right? Now, these membranes are pulled over the eye because they're meant to protect the eye in case the animal's in a fight with something like a seal, which has claws. Okay, so let me show you. Man, birds have them too. I forgot. Shark. Another cool thing about sharks. It was my first thing about marine biology with sharks. I love sharks. Oh, there's a good shot of it. Oh, there's a perfect shot of it. Okay, so this thing covers their eye to protect their eye from claws and that stuff. But of course, when it does that, they're also blinded. Now, in seawater, those aluminum cages give off a lot of electrical sensation. So you'll often see those white sharks swimming up to the cages, and the bait's hanging off over here, right? So you think the shark's going to go for that. Shark's going for the bait, and then at the last minute, they close their nictitating membranes to protect themselves. They're blinded, and then at that time, since their sense of sight is gone, their electrical sense from the Apulae of Lorenzini that overwhelms them, and they'll bang into the cage because they'll even grab, they'll even bite at the cage, right? So that's why you see that, because of the fact that especially in the saltwater, they're getting that confusion of their senses. I mean, it's a good way to detect prey, no doubt, right? Okay, gosh. All right, let's keep going. The smaller you are, the more you resist sinking. It's called the surface area to volume ratio. Again, if you want to do the math, you're welcome to it. But what we're doing is we're comparing those numbers down on the bottom between the crab larva on the left and this thing called the box crab on the right. And what we're saying is, as that S over V, what that basically is surface area to volume ratio. As that number gets larger, then it's more difficult for you to float. More difficult for you to float, right? See, the crab larvae is a 6, the box crab is a 1.5. The crab larvae is going to float a lot easier than the box crab is. Now, you can increase that S over V number by doing things like adding little hairs and appendages, like the Othona or the Gaussian has done. Both of them have done it. Othona has done it more. Remember, we talked about that in class because Othona is a cold water species. Or excuse me, a warm water species. Othona is a warm water species, so you need more drag because warm water is less dense. Okay, streamlining helps you get through water. Obviously, that's something that any organism is going to need. It's something that any man-made craft pushing through the water needs. It needs to be streamlined. Can you imagine once the first cave people went, oh, box doesn't go so good through water. If you make it into, I don't know, triangle, go better. I doubt they spoke that well, but you get the idea, right? That must have been, whoa, what a dawning moment there. Now, in evolutionary history, organisms figured that out pretty early. It's hard to swim through this thick stuff. It's a lot thicker than air, right? So the best way to do that is to kind of, play with physics. That's how it works, okay? To make it less friction, to decrease your friction through the water. And one of the ways you can do that is by making the point of the hull of a ship, okay? It's not a box. And animals have followed that same thing. I told you in class many times it's called a fusiform shape. It's fusiform. Long and narrow, just like you see here, right? Whether you're a penguin, a turtle, which is a reptile, a bird, well, is a reptile, the mammal walrus, the bluefin tuna, some more than others, right? Some are really like sharks, right? Sharks are going to be really, really fusiform. Sharks are going to be really, really streamlined. Now, so keeping that in mind, do you think, what could you deduce about an organism's feeding habits from its degree of streamlining? What could you say about how an organism feeds based on how much streamlining it has? Hello, hello. What do you think? Did I lose my sound? Are you guys thinking, or did y'all fall asleep? You need to drink some coffee before class then. It can't be that boring. Okay. What's one of the things that if you're a competitive swimmer that you do? Yeah. Streamlining, that shape that you see in these critters. Why would that be, how would that be an advantage, or what could that say about an organism's feeding habits, about what type of feeding organism you are? And what I mean by a feeding organism, that's what's confusing. I'm saying, are you a grazer? Are you a predator? Yeah, there you go. That's what I was trying to get. I'm sorry if I wasn't making it clear, but yes. So it's the detective work again. So oftentimes we look back through the fossil record and we say, Animus extinctimus, whatever it is, right? It's no longer around. We could say it was a predator. And you go, how the heck can you tell it was a predator? Predation is an act, okay? So sometimes they actually do, and the fossils catch them. It's really very cool. Like they catch organisms that died right in the middle of eating another one. They even have they even have fossils of organisms that had young inside of them, internal developers that became extinct. So you get these fossilized critters inside these bigger fossils. It's awesome, right? But that's how they tell. They go, oh look, it's very shark-like formed. Why would you be shark-like formed? Because you're chasing stuff down to eat it, okay? And then down below, we talk. One of my most incredible things I love to talk about in evolution is convergent evolution, because it emphasizes or illustrates no better idea that I try to put into my biology students, in that the organisms, what it looks like, everything about it is shaped by its environment. Okay, that yes, a lot of the way you are is your genetics, but ultimately all of that is going to be shaped by the environment. So whether you came from a shark lineage, reptilian ichthyosaur lineage, or a mammalian lineage, which if you go back to that chart at the beginning before I started the recording, yes, we're all connected, but we're always saying, a most recent common ancestor. And fish, reptiles, and mammals all have a common ancestor. They do, but not a very recent one. These organisms look the way they do, not because they're related. I mean, think about all the fish, all the reptiles, and all the mammals that don't look like this. There's a lot more, right? So they look like this because they've converged on the same way to deal with life. That's why we call it convergent evolution. Okay. I think that's the last thing we talked about. Let's go on to new stuff. I can go on about this stuff forever, so we got to move on, though. We got lots to talk about. Okay, one of the things about living in the ocean that's beneficial or might be good about living there is the fact that it's a fairly stable environment. It's a really big environment, and it doesn't really change a lot. Now, I used to work with this woman at SeaWorld. She said, change is bad. Every time they would get rid of a VP and hire another one, she'd say, oh my goodness, change is bad. And I'm like, Chris, you don't know that. But anyway, organisms, change can be bad. Now, for all of life on Earth, change is good because it's created all the different kinds of life on Earth. If there were no differences on Earth, if the Earth had never changed, then everything would always be the same way. So while change is good for kind of the Earth, if you will, or the diversity on Earth, it might not be good for you, because as the environment changes, you might find yourself in a position that stresses you, in which you can't survive, in which your reproductive rate goes down. Now, your buddy next to you, for whatever reason, maybe in this new stressed environment, he's not as stressed. He might actually be stressed just like you, but not as much, and therefore he can reproduce more. Now, one of the things about living in the ocean that's great for organisms is the fact that it does not change a lot. Okay, it's really big, it fluctuates. You know, it's daily, seasonal, annually. Okay, it does. If a storm moves through, you've all been to the beach, and you know that, you know, the water temperature, you can watch the news at night and see the water temperatures. But remember, a lot of time that's going to be right inshore, the very shallow water. And while organisms are typically found in shallow water, we're talking more continental shelf shallow, not, you know, waist deep shallow necessarily, right? It's too rough there. So we say that the deep ocean, way out there in the deep ocean, where there's not a lot of organisms, is what we call isothermal. Iso is a cool little root word to try and remember, because you see it a lot. Like, for example, when we were all in geometry, which I was asked to leave several times from geometry from my geometry teacher. Him and I just didn't get along. Anyway, I do remember this, though. If you put a line, it indicates that the length of the triangle is the same if it's the same number of lines, 1 or 2. So this says that these two sides of the triangle are the same length. And there's a name for that type of triangle. Does anybody remember the name? What kind of triangle is this? Like, this is a right triangle. There you go. Isosceles, the two sides. The two sides are the same length. Right. So iso means same, and thermal is an easy one, right? What's thermal? Yeah, so it says that the ocean, especially out in the deep, doesn't change a whole lot, right? And you can see, if you look at that little chart, that really shows you. On the open ocean, you got a range of, I don't know, 34 degrees, right? And then as you get in closer, okay, as you get in closer, you're going to get shallower water. And shallower water heats up and cools off faster. It's less water, less mass, okay? But it's still not like land, right? In land, you can have, you know, 146-degree differences. Not in one place in one day. Oh my God, can you imagine? Oh my God, it's minus 90 on your way to work, and then when you're on your way home, this is in Celsius too, by the way, 60 Celsius, which 60 Celsius would be, 40 is like body temp. 100 is boiling, so 60 is like, what, 150? Good God, can you, and then minus 100, so it's going to be like minus 120 or 150. How do you dress for something like that? But what we're saying is over all of land, right? Over all of land. And even in some areas, the temperature in a day can change, or even seasonally, quite a bit. I mean, you know, I grew up in, or not grew up, but I was born in Spokane, Washington. And during the summertime, it would get 105 during the day. They're really hot. We could also have a blizzard in the wintertime. And in Seattle, I remember in the wintertime, sometimes even having to put on the heat in the house. So you can get those daily changes. All right? But the ocean, if you're living out there, especially the deeper you are, is a good insulator. It works, those 4 reasons I have listed there, for the same reason many of the reasons anyway, for the same reason the radiator in your car works, folks. The radiator in your car uses liquid to cool off your engine. Now, in the old thermodynamics and the theory, what's going on is the heat is being transferred. It's going from your engine, the metal of your engine, to the liquid, to the antifreeze. And then the antifreeze goes to the radiator, and the radiator, because you're moving through the air, cools that stuff off again. So again, now the heat moves from the liquid, your antifreeze, out into the atmosphere, the air, as you're driving through it. So what we say that that ability of water to do that, and antifreeze is more than just water, but just water of any liquid can do it. Antifreeze is just better at water than doing it. has what we call a very high heat capacity. Antifreeze has a higher heat capacity. That's why you use antifreeze instead of water in your radiator, because you have to get your temperature hotter before it boils. Once your engine coolant, antifreeze, starts boiling, then it all boils away and you can't keep the engine cool. So water also has a very high heat capacity. It can absorb a lot of heat. Water has the ability to absorb a lot of heat, we say, before it changes state, before it boils. States of matter: solid, liquid, and gas. Okay, so before liquid water becomes gaseous water, you got to boil it. And that takes a lot. You've ever put, you ever put water in a microwave and try to boil it? It takes forever, doesn't it? Seems like it takes forever. And what's that old saying? A watched pot never boils, right? That means because if water takes forever to boil, it absorbs a lot of heat before it boils. There's a reason why, right? You know why? There's a reason why with the noodles it boils a little faster. It's the salt. The salt makes it. Maybe put salt in water to boil it, make it boil a little faster. It lowers its boiling point. It's a chemical thing. Okay, so the ocean, and of course, when the ocean is trying to heat up, there's a lot of ocean to evaporate, and evaporation removes heat. So you see what I mean? The ocean is mixing. So for the most part, the ocean is very stable in temperatures. So organisms, especially in the deep ocean, are going to be very weird and freaky and look a lot like critters that did millions of years ago, because ain't much changed around the old habitat there. So, and Mother Nature is beautiful about, if it ain't broke, don't fix it. If the world is, if your environment stays the same, there's no reason to go through evolution. There are some organisms that do live in areas, especially like intertidal areas or estuaries where the tide goes in and out and that sort of stuff. They have to deal with differences in temperature, right? As the tide goes out, the tide goes in. So these guys, these organisms can either be what we call stenohaline or euryhaline. Steno means that they are intolerant of wide temperature variations. So, you know, they live typically in the open ocean where the temperature is not going to vary a lot. Euryhaline is organisms that withstand large temperature variations. They can deal with big fluctuations. They're going to live more in the coastal waters. That's why coral get into trouble. Coral are really kind of stenothermal. They don't really like big changes in temperature of the water. But since they live with this organism that needs to photosynthesize, they have to live in shallower water because they need the sunlight. So, yuck, robbing Peter to, no, rock and a hard place. That old saying, either way, I'm kind of living on edge here. That's why they're so sensitive. Okay, so another thing that organisms have to deal with in the world, not just in the ocean, is what to do about salt water. We all know salt water is drying. So let's talk a little bit about some of the chemistry and how that affects critters. Specifically, we're going to be talking about salinity, but in general, I'm going to be talking about the way things move around in the environment and across your membranes, for example. The easiest one to understand, and that's why it's probably called simple diffusion, is that things like in the picture you see up above go from where there's a lot to where there are few. So they spread out. So the initial state, the molecules of whatever you're talking about, could be dye, drops of red dye, are concentrated in that little drop, and after a while they spread out. And if you come back in an hour or so or whatever, the entire container will be all evenly colored with red dye, right? That's simple diffusion. That happens naturally, normally. It's because molecules bang into one another at the very molecular level. If you've ever done that experiment in lab where you take India ink, which is actually little particles floating in water, and you look at them under a microscope, you can watch them jitter around, and you can't see the molecules, but they're jittering around because molecules are bumping into them. It's just, wow, very cool. So that's simple diffusion, okay? And that's how, for example, you get oxygen into your lungs. You breathe in the air, there's more oxygen in your lung, in the passages in your lungs than in the bloodstream of your lungs, right? So the air goes into the bloodstream of your lungs. Now that oxygen air that you need, that oxygen goes around your body, and your cells are using oxygen, so they're running out of it. And because there's less in your cells than in your bloodstream, the oxygen goes from your bloodstream to your cells. Simple diffusion. I always say things flow down a gradient. Think of a gradient as like a hill. They flow downhill. They flow from where there's more to where there's less. Now, osmosis is a little bit different. Osmosis is still diffusion, but it's specifically the diffusion of water. And in order to understand the way the water is going to move, in order to be able to predict which way the water would move across an organism's membrane, we have to include a discussion about what is dissolved in the water. So take a look at this picture down here, and we have a selectively permeable membrane. Folks, that's just a fancy word for your cell membrane. And we'll call over here side A, and this will be side B, so that when I'm talking, you'll know what I'm talking about. Okay, so let's imagine that side A is the inside of your cells. So this is the cytoplasm of your cells. This is the area where all the metabolic reactions are going on. And the bloodstream, in just sort of a large way, what we call a gross anatomy way, is going to be represented by side B, your blood vessels, your bloodstream. That which is bringing you the nutrients, that which is removing waste, right? Carbon dioxide is waste, so your cells make it, goes into the bloodstream, and not only do they deliver oxygen, they get rid of CO2. All right, now, so you understand what I mean. Inside the cells is A, out there in the blood vessel is B. Now, those green things that you see there in this particular picture would be sugar molecules. They could also be salt molecules. They could be protein molecules. The gist of it, or what I'm trying to get at, is that what happens when you put molecules into water is water sticks to them, kind of like if you dropped a lollipop at the beach, right? Like a Tootsie Pop, and you dropped it, oh man, into the sand. If you picked it up, it would all be covered in grains of sand. Think about the lollipop as being maybe a protein molecule, one of these sugar molecules, and the grains of sand as being these water molecules stuck all around it, okay? You get that visual? Now, the selectively permeable membrane is called that because it's got little channels that basically, in the simplest form, you got to be small enough to fit through. So that's why the sugar molecules are too big. They won't fit through it. Right? Now, look at how some of the molecules inside B, and some in A, but more inside B, are attached to the sugar molecules, and some of the molecules are not attached. Now, we're going to call those two different names. We're going to say the ones that are attached are going to be called the bound water molecules, and the ones that are unattached are going to be called the unbound. See what I mean? These are the bound. These are the unbound. Let me go back and redefine osmosis for you, and I think it's going to help you now understand. What happens in osmosis is the diffusion of water, okay? But add one word between of and water. Add this word. It's the diffusion of unbound water molecules. The bound water molecules, once they're bound to sugars or proteins or salt, can't move. They're too big. The molecules of sugar or salt themselves can't move. The water molecules stuck to them can't move. So the only thing, Mother Nature is trying to balance the water here. So and this is laws of physics, folks, it's called osmotic pressure. So the only thing that can happen in this case is water molecules are going to move across to balance themselves in terms of the unbound water molecules. Now, the arrows might be a little confusing because you got some molecules going one way and some going the other. What we're saying here, notice 3 arrows are going to the right, though, right? What we're saying is water is always flowing across the membrane, back and forth, but there's going to be a preference or a tendency. Now, put this in your notes because you'll never screw this up if you remember this one little phrase. Water, unbound water, is always going to flow to the side of the membrane that has more stuff dissolved in it. All right, that has more salt or more sugar or more protein. That's the way the water is always going to go. Okay, so if I were a cell and let's say that I had 1% solute. All right, solute. What's a solute? A solute, I'm typing this, by the way. A solute plus a solvent equals a solution. water diffusion of unbound water molecules. Water always flows to the side of the membrane with more stuff dissolved in it, more solute dissolved in it. Okay. So a solute is the stuff that was getting dissolved, like in sugar water. Sugar is the solute, water's the solvent, the solution is sugar water. Okay. So in osmosis, to use that word now, osmosis is the diffusion of unbound water molecules to the side of the membrane that has the higher solute concentration. Okay? Now, here's a cell on the left. Our cells, in general, are about 1% solute. About 1% solute, 99% water, okay? I put you into the ocean. You go swimming at the beach. The ocean is, if you remember from one of our earlier chapters, about 3.5% salt. Which way is the water going to go? Is the water going to go into your cells or out of your cells? Which way is the water going to go? In or out? Yeah. Ace says the water's going to go out. That's right. Water's going to go out. Now, let me erase all my scribbling. Answer me this. There we go. Let's go back to the same cell. I'm going to use 1% again because, again, animal cells, plant cells are all about 1%. Let's say I put you into distilled water. Now, distilled water has nothing in it. It's 0%. Which way is the water going to go? Okay, very good. Now, final one. Erase the 0. What if the concentrations are the same? Perfect. Perfect. Now, to be technically correct, water will flow, but it'll flow across the membrane equally. So for every water molecule that goes out, another one flows in. See, in the picture, you remember, there's more sugar molecules on the right, so the net flow, there's 3 red arrows going out. Okay, does that make sense, everybody? So organisms, just like in thermal, can also be stenohaline or urohaline, whether they can deal with big concentrations of salt change. That's going to be organisms that live, you know, in the shoreline, in estuaries, areas where the, you know, where the tide goes in and out. And if you're out in the deep ocean, you're going to be stenohaline. You're not going to be able to deal with those are wrong. Holy cow. This is, steno is small. That's open ocean. Large, sorry, it's an error. Let me change that in my slide. I didn't even notice that. If you wonder, like, what in the same definition? All right. Okay. So let's take a look at this next chart just to kind of emphasize that one more time. I got an animal cell and I got a plant cell. So an animal cell, fish, or anything that lives in the ocean. And all around this is the what we call the extracellular environment. So again, it's like the beaker. So if it's an animal cell, you told me that what happens is the water is going to flow in an environment that we call hypotonic. Okay, now tonic means solute. And hypo means less. So that word literally means less solute. Now, again, that word is referring to the extracellular environment. So the water, as we know, flows into the cells. Because if you're in a hypotonic environment, the environment has less solute than you do, the cell. So water is going to flow into the cell. Now, if you're an animal cell, that's a problem because you're going to burst. Okay? You're going to lyse. A plant cell can do that, and plant cells like that because plants, especially ones on land, they need to save water. You know, if it gets dry out, they can't walk over to the next river and get water, so they're going to save water. So they actually have what we call a cell wall. It's made of cellulose, cardboard kind of. Well, it is cardboard, it's paper. And that cell wall allows plant cells to really fill themselves up with water. It's like blowing a balloon up in a box. Blowing a balloon up in a box, you could never explode the balloon. The box would hold it in shape. Skip the middle one for a second, but go to the other side. Hyper now means higher. So higher solute. So now you're in the ocean. In the ocean, water is going to go out into the ocean from your cells. You're 1%. The ocean's 3.5%. So let's go with 0% when you're hypotonic. 1% here. Plant cells are about the same. This, remember, would be like, say, for example, the ocean, 3.5%. Still 1%. Okay, so the water's going to be lost. In animal cells, it's called shriveled. Another word for this you might see sometimes is called crenate. Crenate means to have irregular edges. A steak knife would have a crenate edge. Yeah, some of it is. Did you take Bio 1? Cool. Do you remember this? Do you remember these terms? Cool. Well, you're not going to remember the first time. That's the nice thing about seeing these things over and over again. I think I probably learned the steps of mitosis, which you learned in Bio 1. I bet you I saw those steps of mitosis 10, 12, 15 times all my years of college. I saw it in, biology. Then I saw it in genetics. Then I saw it in advanced genetics. Then I saw it in evolutionary genetics. So just because you saw it once. All right, then I'll leave you alone as far as that goes. How about isotonic? Which way is the water going to go? It's going to go in and out, isn't it? But it goes in and out equally. Plant cells, we call that flaccid. That's okay for them. They're not upset with that. They definitely don't want to be this. I'm sorry, backwards. They do want to be this. You and I don't want to be this. We don't want our cells to do that. We want to be this. Do you see my happy faces? Hopefully I'm not making this more confusing. And even plant cells don't want to lose water. What is with me doing that? You see what all these scribbling on the page? When I was in college, that's what I do to my notes. I would write all these stupid little faces and all this kind of weird stuff to help me remember stuff like that. Years from now, they go back and if they find my notes, they'll say, oh, there was some sort of secret code. It was just me scribbling to help remember stuff. Okay, so if those you added in Bio 1, now let's apply it to what we know about marine science. Here we got, or let's go with both kind of fish. Let's not just exclusively marine fish. Let's talk about freshwater fish first. So let's talk about fish that live in a hypotonic environment. Now, ignore this. The word hypo and hypertonic are relative. Okay, that's why sometimes you see the words reversed. I'm going to say this just once. You can forget it if you want to, because every time I refer to those words hyper or hypotonic, I'm going to talk about the environment. But let's say, for example, what we're talking about is a hypertonic environment. In a hypertonic environment, the ocean has more solute than you do. Okay, so the ocean is hypertonic to you. What are you to the ocean, though? What are your cells to the ocean? Are they also hypertonic? Cooper snoring in the background. It's 3 o'clock in the afternoon, and I'm thinking, oh my goodness, the blood sugar is dropping in y'all's brain. I know exactly what that feels like. Yeah, you got it, Ace. So if something is hypertonic, if you're hypertonic to something, that something is hypotonic to you, right? If someone is losing water, someone is gaining water. Enough, right? Okay, so I'm always gonna be, so the freshwater fish, regardless of what it says up here, and what it says is not wrong, I'm just gonna try alleviate confusion. If I'm talking about a freshwater fish, freshwater organisms are found in hypotonic environments. The lake is hypotonic. Saltwater fish are found in hypertonic environments. The ocean. Okay, so what happens to a freshwater fish in a hypotonic environment, okay, well, in a hypotonic environment, water is going to flow in. See, water gets absorbed by osmosis. How do they deal with that? Okay, for one thing, because you don't want to absorb too much water. Goldilocks, everything in the middle, huh? You don't drink. You have special glands, oftentimes in their gills, that actually help them secrete salt, okay, and they also can lose salt through their skin, and you pee a lot, all right? You urinate a lot, okay? The other way around, if you're hypertonic, then you drink a lot of water. Well, that doesn't help because guess what? That water is salt water. But you have gills, much like the skin and in some of the gills of freshwater fish that secrete the salt. So you drink the salt water and take the salt out and get it out through your gills, and you keep the water. And you have to do that because you're going to be naturally drying out, and you don't pee very much. When I was in graduate school, what they do is that you give yourself what's called an oral comprehensive exam, and that means your professors on your committee. There were 3 on my committee, 4, and what they do is they sit you in a room, and for as long as they want, they ask you whatever question about biology, from your general biology 1 days all the way up to the last course you had when you were a master's student. This is where you're getting your master's degree. And what they usually do is they start out simple, and then they get more complicated, and then they find out where your level of knowledge is. And it's as nerve-wracking as it sounds, okay? I was in there for 2.5 hours where they basically just orally ask you questions. And I was a smart aleck, and I thought that I would schedule it over lunch, because they tell you you can schedule whatever you want. So I said, all right, I'll start it at 11. Figure by noon, 12:30, they'd be hungry. They were. They brought out their lunch and started eating it in front of me. I didn't have my lunch. First question my professor asked me, or he was the one to lead off, my major professor said, okay, I take a fish out of the ocean and I throw it in the lake. What happens to it? Could you answer that question? I take a tuna. I take a fish out of New Smyrna Beach. I throw it in one of the lakes in Lake County. What happens to it? Osmosis-wise. Yep. Why? What would happen to his cells that would make him die? Yep. This is exactly what would happen. Got that question right. Didn't get them all right, but got that question right. So if you ever go through grad school, it's an interesting experience. Oh, okay. Great question. Tuna, sharks, they all do that. Oh, I'm glad you said that. I didn't want to forget this. Here's what those organisms do. Blah, blah, blah. Where do I want to talk about this? Right here I do. All right. If you can somehow adjust the amount of solute on either side of those blood vessels, all right? So for example, if your blood becomes salty, if you can somehow deposit more solute into your cells, and likewise, if your blood becomes less salty, if you can get rid of that excess solute, so you can somehow pump solute in and out, you can always remain isotonic. That's what they do. Sharks and tuna, sharks use their urine. They use uric acid or urea, and they store it in their bloodstream. That's why folks who fish for sharks will always tell you the important thing about a shark is to bleed it well, because if you don't, the urea, the urine in the blood will get into the meat, into the fish, and make it taste bad, make it taste very ammonia-like. You can always tell that. If you buy shark from someone, smell it. If it smells like ammonia, then it was not bled right. You're right. And the way they bleed it is you cut the tail of the animal off when it's alive, so its heart's pumping and it gets rid of all the blood. Anyway, so the point of all this is that's how they live. These organisms, and oh my goodness, there was a shark. It's actually, they called it the Lake Nicaraguan shark because it lived, they thought, in Lake Nicaragua. But Lake Nicaragua is connected by the river, I can't remember, a river that's connected to the Caribbean. So these sharks weren't living in the lake. They were swimming up the river, and the reason they were doing that is all the natives around the lake would come down and wash their clothes and get water and use the lake, right? So these sharks were just polishing these people off. And they were like, where did these sharks come from? And sure enough, they were swimming up the river. So, and they do it in Florida. You'll find bull sharks out in the Indian and the Banana rivers, and those are brackish. Those are like 15% salt, not like 1.5% salt, not like the ocean. So if you can somehow keep, and that's what these animals do, if you can somehow keep that solute constant and vary that somehow, you take my Bio 2 class, I'll tell you about the rectal gland. Sounds awful, doesn't it? Rectal glands. You know what? Let's look at a rectal gland. It's not quite as gross as it sounds. Yeah, if we were to dissect a shark, and please, the word is dissect, not dissect, but if we were to dissect a shark, near the anus of it, we would find this little tiny organ. It's called a rectal gland. That's what sharks use. That gland secretes or absorbs solute to keep themselves isotonic, no matter what kind of environment they're in. Awesome question. Thank you. Thank you. Any other questions before we go on to this one? This is another incredible, incredible little piece of machinery. All right, how fish breathe in water and things with gills in general, and actually things that have thin enough membranes. In biology, that any membrane, okay, I was going to say skin, but that's not really right, but any membrane that's thin enough and that is vascularized. Vascular. Now, what's that mean? Vascularized. Your where the inside of your nose is highly vascularized. Amy, when you got your nose pierced, did it hurt? Is Amy still on? Hello? Oh, yeah. That's kind of like when they did the top of my ear. I was like, ow. I took him out eventually. I was like, man, that hurt a lot more on the bottom of my ear. Any area that's got a lot of blood vessels, though, okay, any area like that we call highly vascularized. And if it's thin enough, it can actually, gases can diffuse across it. Carbon dioxide and oxygen can. That's what the gills of fish do. On the right are the gills of some fish. Look, they're highly vascularized. You can tell that because they're very red. And they're very red because they have lots of blood in them. Now, let's look at the picture up above first, and then we'll talk about the picture on the left. The picture up above is the large view of how fish breathe in water, okay? They open their mouth. They have these gills on either side, and they have gill slits. Sometimes they have a covering. That covering is called an operculum. Right, there's the word operculum. All right, so on their inhalation, they close the operculum, okay, when they're bringing in water, and they open their mouth. Now, when they open their mouth with their operculum closed, physics takes over. There's no water in their mouth. There's lots of water in the ocean. Water goes from where there's a lot. Now, this isn't osmosis, folks. This is just stuff. This is diffusion. Water flows into the mouth. The animal closes its mouth, opens its operculum. Oftentimes, what they do is they'll have a tongue, and they raise their tongue to the roof of their mouth, squeezing the water out their gills. Okay? That's how they filter feed as well. Because one of the things that are on gills, you can almost see them on the front of these gills right here. Those are called gill rakers. Gill rakers keep the gills clean, but what they also do is fish can use them to trap things, to trap food on, okay? You can see a gill raker in the illustration up here. All right, now, I don't want to get too fixated on the terminology, but the little sheets of vascularized tissue, those are called lamellae. Lamellae means thin paper-like slivers, okay? And the lamellae are made of gill filaments, and I want to kind of look here mostly. The really cool way that fish receive oxygen from the water uses diffusion. You see, when their gills are open, the red arrow that you see, that's the way the blood is flowing. And the blue arrow that you see, I'm looking at the picture, the bigger one on the right, the blue arrow is the water. So it's called a counter current system. for reasons that make sense, right? The blood is flowing one way, the water flows the other way. Now what does that do? Well, now look down to the lower left. Imagine that on the right-hand side is the capillary. That's your blood vessel. And on the left-hand side is the water that's moving over your gills. Now, they're moving in opposite directions. So your blood vessel, you're receiving deoxygenated from the blood, blood from the body. You're receiving blood that has already been used by your body. There's not a lot of oxygen in it. There's not a lot of oxygen in the water that's flowing over that part either. But diffusion doesn't say how big the hill's got to be. It just says there has to be a hill. So that means there's going to be a net flow of oxygen from the water into the gills. Now, that gets better as we move further away from the deoxygenated blood. So as the blood becomes more and more oxygenated, well, the water too is more and more oxygenated. So as one concentration increases, okay, as the oxygen in the blood increases, the oxygen in the water increases. So it's always flowing from the higher to the lower. Don't confuse by me calling it the countercurrent as the fact that the concentrations stay the same. The countercurrent is the blood flows one way and the water flows the other way. The picture down below using diffusion explains why this works. Is that cool? Oh man, we might end up having, I got plenty of time in this semester though. I'll have to ask you guys lots of questions on the exam about this. Since I'm spending, am I supposed to be on 12? I am on 12. All right. You guys don't mind if I talk about this stuff in detail, do you? I love talking about this stuff. Any questions you have? Okay. All right, good. Coloration. Living in the ocean is a hairy place. All right. There's lots of things that want to eat you. Lots of things you want to eat. You want to hide from things that want to eat you. You want to hide from things you want to eat. Okay. So you want to either get away from being preyed upon, or you want to find your prey. So there's a couple of ways, and a couple of really unique ways that these critters can do it. For one thing, like the jellyfish, you can be transparent, or like the Leptocephalus, right? You can be transparent. You can hide. Can you find the leafy sea dragon in the middle picture? I guess I should have had a bigger picture of that. But can you see the sea dragon? The sea dragon is a seahorse right there. They like to hang out in seaweed. Can you imagine why? The fish to its right is called a sargassum. Sargassum fish like to live in sargassum. Sargassum is a weed that you have found on the beach in Florida many times. If you've ever gone to the beach, then you've seen sargassum. Here is a picture of it. Sometimes you might have even seen thick amounts of it. I always tell my students in Bio 2, it's easy to recognize sargassum. It's very curly, so there's a lot of S's in sargassum, a lot of U's, a lot of curly letters, okay? The fish on the right is a sargassum fish. Sargassum fish live in sargassum. Can you see why? Doesn't it look like a piece of sard? Now, these are vicious predators. We caught one time on a cruise. I was on a scientific cruise. We brought it back to the laboratory there at UCF, and we used to eat little bits of hamburger. I mean, this thing didn't mess around. So while it was hiding from stuff, it was also using this to sneak up on stuff. Camouflage, counter shading. Look at counter shading. Look at the white shark, killer whale, and then for the halibut, look at the halibut. Oh, you know what I said? For the halibut, look at the halibut. All right, take a look at those. That's what we mean by counter shading. Dark on top, light below. Dark on top, light below. Dark on top, light below. If I'm above you, it's hard to see you looking down. If I'm below you, it's hard to see you looking up. It's lighter above you, darker below you. Killer whales, sharks, halibut, you got one fish here a very different type of fish here, and holy cow, a mammal there. And they all come up with the same answer. Again, that convergent evolution, right? Works really well. Works really well. That's why oftentimes those surfers out in California that get hit by white sharks, and it happens to be a white shark, don't know it's coming because they're looking at them, they don't see it coming. They're looking out the gray. If you're a seal, wow. For the shark, that's a great advantage. Camouflage the rockfish down here too. Rockfish for names that are quite obvious. Warning coloration, it's sort of the obvious or the opposite of camouflage. Now you want to stand out. Now you're basically saying, you want a piece of me? You know, you got a t-shirt on that says, you know, bad things about your mother, right? It's like these fish say, I dare you to come touch me. These organisms say, I dare you to come touch me. Those spines on the lionfish are venomous, and they're found all over Florida now, unfortunately, because they've been introduced, right? They're saying, don't screw with me. Now, what that basically is remember. Remember, it's like, oh, don't mess with them. Okay, so that's a very effective deterrent. It gets magnificent amounts of pressure at the bottom of the ocean, right? 16,000 pounds per square inch. There's the little formula. For every 33 feet, for every 10 meters, it increases another 15 pounds. How do critters live down at the bottom? Well, I always make a joke and I say, well, there's a lot of flatfish down there. And it's kind of a joke, but it's kind of not. A lot of marine species, the way they deal with deep water is they don't have things that can be compressed, right? They don't got lungs. They don't got ear canals. They won't have a lot of body space. So there's not a lot of things that can be messed up by being mushed together. A lot of really deep water fish are very flabby. Like even if you catch them like fresh, they feel like, this thing's been sitting on the floor for a couple of days. It's a really flabby, what they've just done with their bodies, they don't even just fight back. They're like, oh, just mush me all over. I don't care. I think I told you guys about before. Look at the pressure, right? This is a styrofoam cup, and these are the ones that they brought down on some of those submersibles. Just squeeze the air right out of it. But if you don't got those spaces like these fish don't, like these fish have in many marine mammals, then you don't have to worry about that. Fish have a unique structure called a swim bladder. Swim bladder is right about in the middle of the fish. Now, it looks like it could be a lung, and the evolution of lungs might have actually came this way, but it doesn't work in exchanging gases. That's going to be the gills. The swim bladder is a buoyancy thing, okay? The swim bladder up above, I got a better picture, fills up with air. More air, you float up. Less air, you float down. If you're a certified scuba diver or if you've ever gone that way, you know that's what you do with your BC vest, right? You inflate it with air or anything. You've been on a float. Now, it works by being connected to the bloodstream. This thing called the oval body absorbs the air, so when it wants to remove the air, and the gas gland releases the air. And the mechanism by which it does that, Mother Nature, she's so brilliant. Don't make things the same way twice. It's another countercurrent system. So it works almost exactly well. It does essentially, in the physics level, work exactly like what's going on in the gills. Oh, man. Years ago, when they were first examining the bottom of the ocean with sonar, they found this weird false bottom. There's a picture of it right there called a scattering layer. They called it the deep scattering layer, DSL. The problem, well, not the problem, but the really weird thing about that is this thing went up and down. This layer of whatever the heck it was, can you imagine the first people? Like, that's surely a sea monster, right? So they got this thing that's going up and down, up and down. Now, what it was caused by, it was caused by our friends, the plankton again. So this very dense concentration of plankton was actually dense enough that it was returning an echo signal. So that's what was causing that line that you see on the sonogram there. Now, what these plankton do is at night they come up. They come up at night to escape the predators, you know, and then during the day when the lights come out, then they go back down to the bottom, right? So a deep scattering layer is basically just kind of staying one step ahead of these different types of predators, because there are daytime predators, which are called diurnal predators. Nocturnal predators are nighttime, and this is a cool word. I like this word, crepuscular. Crepuscular, like rabbits are crepuscular. They come out at dawn and dusk. So they come out at, just as the sun's coming up or it's going down. I like to say that crepuscular organisms many times are prey species, like rabbits, and they'll do this because what they're trying to do is catch the predators in shift change. There's the night predators, there's the day predators, and I always kind of think about, good night, Ralph, good night, Joe. People are going, the day predators or the day predators are going home, and the night predators are coming on, and then it reverses in the morning. It reminds me of an old cartoon, which I don't have time to talk about. You guys probably don't remember, but okay. Now, there's not a lot to say here, and I'm pretty close to finishing the chapter, and I have one more slide I really want to go through, but very quickly, the ocean is divided into zones, right? And those zones are based upon the environment, and so we've given names to them. So there's the pelagic zones, and the zones within the zones are oftentimes broken down by large environmental characteristics. Like in the pelagic, we got, do you have a lot of light, a little bit of light, or is there no light? So you got the epipelagic, the mesopelagic, and then down there you got the bathypelagic. So I put it in underline. Euphotic, photic is light, dysphotic, and aphotic. Photic is light. When you put an A in front of a word in science, it means the opposite of. When you say dis, it means sometimes not enough or a little bit, and U means a lot typically, or true. Most of the marine biomass, as you can probably imagine, is going to be in the euphotic zone, right? Because that's where most of the photosynthesis is happening. We know the dysphotic zone begins because it's about that time when the dissolved oxygen, that's what DO means. When the dissolved oxygen begins to decrease at about 200 meters, we know that the reason why that is because the bioluminescence is dying off. Or excuse me, not the bioluminescence, the photosynthesis is dying off. Photosynthetic critters are becoming fewer and fewer because you're getting deeper and there's less sunlight. There's a lot of bioluminescence down there. I'll show you a picture of that in a second. This is where we reach this thing called the oxygen minimum layer. And I told you about this one in the earlier chapters. At about 700 to 1,000 meters, the oxygen levels that had been dropping, and they've been dropping because of the less photosynthesis, because you need light for photosynthesis and you give off oxygen. They actually start to go up after that depth, and that's because all of that water down at the bottom is polar water, and that usually has a lot of oxygen in it. And then most of the oceanic, what we call the oceanic province, is actually made up of the bathypelagic, the really, really deep stuff, in the abyssopelagic. Critters like this. Check this out. Look at the fish in the upper left, okay? See that fish? This is a fish that's found in those mesopelagic and bathypelagic. That fish's eyes actually have two retinas. It can see above it, and it can see to the side of it. Where the predator is going to come from in deep water, they're not going to come from any deeper. I'm already living down where it's dark as it can be. They're going to come from above me, or they're going to come to the side. And bioluminescence and what we call counter illumination, if you can light yourself up, that's a squid, by the way. If you can light yourself up, you can maybe freak your predators out, or you can fool them. Predator's like, I don't know what that is, and it stays away from you. Some species, like this little species of fish, they use bioluminescence to recognize one another. You tell the different species of these fish, these little what they're called myctophid fishes, by looking at their bioluminescent patches on them. See, right along the fins there? So males and females will recognize each other that way. Sometimes, like these anglerfish, oh my God. Anglerfish are called anglerfish because they fish for other fish. They're down there in the dark. They got these really terrifying looking teeth, and they got these bioluminescent bacteria that grow in this thing called an esca. It's a little glowing thing at the tip. And a little poor unsuspecting fish comes up and goes, what's that? He's eating. And speaking of eating, you're going to make your jaws distendable. You're going to make big distendable. That's like disconnectable. Is that even a word? I think so. Distendable jaws, because you're going to want to be able to eat anything. Remember that you got this nutrient rain that's coming down. You want to be prepared to pretty much eat everything. So those jaws better be able to accommodate pretty much any size of anything. Some fish, right? Like these fish, these are goosefish. Goosefish, the females are the fish, right? They haven't really fish. And the male is a little tiny parasite that's attached to her. Males are born without the ability to feed. So they basically, as soon as they're born, they've got to find a female right away. And they clamp onto the female near her what we call the cloaca. The cloaca in a fish is where, it's actually Latin for sewer. A cloaca is where the urine, where the reproductive organs, the reproductive cells, the gametes, it's where the defecation is all going to come out of. But that male parasite will attach himself back by that cloaca and actually become part of her tissue, become parasitic. He'll grow into her tissue and receive nutrients from her. And the only thing he's got to do is when she spawns eggs, he deposits sperm. What a life. What a life. You know, you got those first few moments of anxiety. You've got to find a female. And the reason why you do this is down there, it's dark. So when you find somebody, don't let go. Don't ever let go. You can't be going out or trying to find another mate, bumping around in the dark. Yep. All right. Oh, you know what? I'll finish these last two slides. Again, just a number of definitions. The benthic is going to be the bottom. All right. So the benthic is the ocean floor, and that's broken down to what we call the littoral, almost like literal. The littoral is high tide, low tide. That's why it's called intertidal. The place up above, the spray zone, all right, that's the supralittoral. The sublittoral is the low zone, or I'm sorry, the lower tidal zone, and it's broken up into inner and outer. Okay, the division between the inner and outer is important. This is marked by where there's nobody on the sea floor. So this is the depth at which the water gets deep enough that plants that are attached to the bottom. Now, this isn't talking about the plankton out there photosynthesizing in the middle of the ocean. This is the ones attached to the shelf, if you will. This is where that stops, okay? And then finally, we have it broken down up above into things we call provinces. Okay, we have the Neritic and the Oceanic province. Out there in the Oceanic province, we have what we call pelagic environments, okay? These pelagic environments, if it's Neritic, then it's going to be less than 200 meters. So the Neritic province is the pelagic environment over the shelf, and the Oceanic is the one that starts at the shelf. I know, it's like divided and divided and divided, isn't it? So there's the Neritic. It's all about the shelf when it comes to these provinces. Then you also have benthic environments. Remember, benthic is always bottom. And with the benthic, there's going to be the spot that's underneath the shelf. Okay, so these are going to be organisms that are on the bottom of the shelf. And then as you go down, you have the bathy benthic, the abyssibenthic, and down in the hadal. I always remember the hadal is the deepest because Hades is another word for hell. Hadal. And organisms all on Earth are going to be stratified based up and down these different depths, and it's all because of the environment. And again, if you want to talk more about the environment and how it changes critters, you can take my Bio 2 class. That's where I talk about that. Okay, folks, I went a minute over. Sorry about that. Thanks for sticking around. If you don't have any questions, and y'all have a good weekend, and I will see you in class on Monday.